Identify the most enriched functional pathway as well as the genes that are involved. Okay, so this is the most enriched functional pathway. If we want the genes that are involved, we can right click on this. Can we open in a new tab? Yes, and we open in a new tab. Okay, are we seeing what I've done? If we want to see the genes that are involved, we submitted a list of genes, not so. This database has collected the genes with respect to the pathways that are involved and has shown us the pathway that is mostly enriched with the highest number of genes that we submitted. Okay, now our question 7 says identify the most enriched functional pathway as well as the genes that are involved. So if we want the genes that are involved in this highly enriched pathway, we right click on this gene and we open in a new tab. Okay, yes, it's still loading. The new tab is still loading. Okay, so let's continue with the other part of the question. Now, question 8 says, for the gene ontology, GO means gene ontology, identify for each of the following 3 categories the most enriched pathway as well as the genes involved. Okay, the first one is biological process. Biological process, okay? Gene ontology. GO means gene ontology. So let's check whether our window has already, it is still loading. Okay, so since we have arranged our table like this, okay, we want to look for the most enriched pathways for gene ontology. And under gene ontology, we have 3 categories: biological process, molecular function, and cellular component. Okay? So we'll go, the first biological process that we identify, that is enriched, since we have categorized our genes into the most enriched. Okay, so we'll have to come here and look. So this is, when you see this GO term, it means the gene ontology term. Okay, so if we look at gene ontology term MF, this MF means that it is the molecular function, but we are looking for biological process. The first one, we want to look for biological process. Okay, we go down, we have gene ontology term cellular component. That is not what we are looking for now. Okay, so we move down, we are looking for the first biological process. Okay, it is denoted as, just as the name, BP. Okay, so this is the first biological process here. Okay, so this is the first highly enriched biological process. If we go down, we'll see other gene ontology term biological processes. But since they are coming below, it means that they are less enriched. So this is the first, which means it is the most enriched gene ontology term biological process. Okay, so it is transcription and DNA template. Okay, so we can take transcription and DNA template. DNA templated. Okay. Now, the first highly enriched molecular function. We go back and we start coming down. So this is the most enriched molecular function. The same like we did for alternative splicing. If we want the genes that are involved, we can right click here and select to open in a new tab, or we click directly. Okay, so the gene ontology term, the most enriched molecular function is proton binding. Okay, and the most enriched cellular function is cytoplasm. They are confused also, except they tell you what they are saying. That is why bioinformatics is there to canalize the information that you have to give you an interpretable information. From that list of genes, we could not say anything, not so, apart from the fact that we'll go to an experiment and start testing them. Okay? Now, with the pathway prediction, it can try to give us an information that we can even publish. Not so. So, biological process is trying to tell us that those genes might be involved in this biological process, transcription or DNA templating. Okay? Then molecular function. Which molecular function can these genes be involved in? Okay? It's telling us that it's protein binding. So most of these genes that are targeted by this microRNA may be involved in protein binding. Okay? When they are involved in protein binding as the molecular function, what may they be doing there? They might be carrying out transcription as the biological process tells us. So you see how we are interpreting the information. Okay? Yes. Where are you? The thing is that you are following and copying. Instead of trying to understand what I'm saying. Because when you understand what I'm saying, it is easier for you to go from those principles and do what you want to do. Are you at the level, have you already selected for cellular components? Voila, now we are at the level of trying to interpret what this information can be telling us. Okay? For the mostly enriched functional pathway, we have gotten that alternative splicing is the most enriched functional pathway, and also it might be telling us that that list of genes that we collected like that, the microRNA that targets those genes might be involved in that alternative splicing. Okay? Now, what does gene ontology do? It is grouped into 3 categories, and each of those categories is trying to give us a particular information about the list of genes that we submitted. Okay? And I'm saying that for biological process, it's trying to explain the biological process in which those genes or that microRNA may be involved with respect to the list of genes that we submitted. Okay? So the mostly enriched biological process is transcription and DNA templated. Okay? So it is trying to say that that microRNA may be involved in the biological process transcription. Okay? So what is its function there? We come now to its molecular function. Okay? So what could it be doing there? The molecular function is saying, it may be carrying out protein binding. It may be involved in protein binding. Okay? Okay. So, the third one, cellular component. So, in which compartment of the cell is it found? So, it might be likely involved in the cytoplasm. Okay? Voila. It might be involved in the cytoplasm. So, that is how we interpret the data that we have gotten. Are we together? Voila. So, you see now that from that list of genes, we have gotten an information. This information that we have interpreted. Okay? Voila. So that is how you interpret it. So we are done.